Good afternoon, all of you. Today we are going to discuss about biological control, which is one of the component of integrated pest management. So let us see what it deals about biological control. So today's objective is that we will be knowing that integrated pest management, what are the components, and that what is the importance of biological control agent, and what are the types of biological control agent, as well as finally that there are the basic the component, that is the predators, parasitoids and pathogens. Separately we will discuss about the biological control agents in totality. So overall what we see that exploration of eco-friendly pest management tactics such as integrated pest management, it aims at suppressing the pest population level by combining all the available methods and in a harmonious way to emphasis the least emphasis of this chemical insecticides. So the integrated pest management, the component, as we know that it is physical, mechanical, cultural, host plant resistance, then chemical and biotechnological, all this. Whereas the today's emphasis will be the biological control agent and how it could be integrated as a part of integrated pest management. If we define the biological control agent, then Dr. H.S. Smith, he has defined in a very simplified manner that the use of natural enemies for the control of harmful insects, other animals and plants. He is, you can say that he has coined the term biological control in 1919. And the simplest definition what we can give is that study and utilization of biological control agent such as predators, parasitoids and pathogen for the control of injurious insects. Another pioneer of biological control agent is Dr. Diebeck. He has elaborately defined biological control agent as the action of parasites, predators or pathogen in the maintaining of another organism's population density level at which is at lower than would occur in their absence. So this is what Diebeck has defined or elaborated it. So why we want to use biological control agent? Of course, there are several components are there, out of which why we want to use biological control agent. So here the simplest that there are several advantages are there compared to other components which is a part of integrated pest management, such as one we can take that once we are using biological control agent, then the insect pest population will never exceed integrated economic injury level. That is one of the most important point is that if the insect population is increasing, then followed by the population of biological control agent will follow, and it will bring down the population of the pest in focus or action. The next advantage is that it leaves no residue, such as if you use a chemical control agent, then chemical controls, then we see that it leaves pesticide residue and becomes a hazard to the not only the human being but as well as to the environment. So this, if use of biological control agents, leaves no pesticide residue. Such as here you can see that as the pest population is remaining at a certain level and we are introducing biological control agent, then after certain period of time, when after the successful attempt of or controlling the insect, their population will bring down to the level what you call at economic threshold level. So this is one of the benefit of use of biological control agent. Another advantage is that unlike the pesticides and physical tactics, biological control typically does not disrupt other pest control tactics. So, this is you can say that this is it could be integrated in a pest management system. And if it is successful, of course, the initial cost of the use of the biological control agent is much more in case of the research, but once it is successful and it is introduced in a system, then there will be no further cost involved in that, because the biological control agent will always control the insect population which is trying to damage our crop. And of course, it leaves a successful biological control agent is relatively inexpensive. It means it involves no cost and it is highly economical. So, summarizing this, the advantages, we can say that it is a safety is involved. This is the first attribute of biological control agent, and besides, the hazards of pollution and resistance posed by chemicals does not exist with this method of control. Second is permanence. Once the biological control is established, then it will remain for a long period of time and without the intervention of other control methods. The third is that economy. Of course, once it is, as just now we have said, that initial expenses will be more, but once it is successful, then no cost is involved in its regulation. Only disadvantage of use of biological control agent is that it is only slow pace. It takes time in establishment. However, once it is established, then there is no question of you can say that it will remain for a longer period of time. So now, the biological control agent, there are three types of biological control agents exist. One is the predator, another is parasitoids or parasites we call it, and the third is the pathogen. The predator, as we know that it is an animal which feeds upon other animal, in a broader sense what we call it, whereas parasitoid is very small and it is parasitic in nature, and which lives in or a host, and eventually it kills it in the process of its development. Whereas the pathogen, they are macroorganism, and they live, or you can say that they feed on in a larger host organism and cause injury to it. So, in the following we will be discussing a little bit in detail. So, here you can see that categories of natural enemies. One is microbial enemies, such as you can say that predatory and parasitic insects, such as you can see here that this predator is feeding on aphids. Second is another predator is there, and third is parasitoid. Whereas predatory vertebrates, you can see here that myna is there, then lizard, and then mole, and then even the the vertebrates could also be used as biological control agent. But there again there comes that the production is one of the problem, which that's why we are not using that predatory vertebrates. However, it could also be used. Next category is microbial control, which contains the parasitic nematodes, protozoan diseases, parasitic fungus, then bacterial diseases, as well as the viral diseases. If you go to history, that first Chinese in 1900, they have used the faro ant for the control of leaf eating caterpillars. Then in 1602, Al-Rawani first introduced this hymenopteran parasitoid, the name is Aphelenterus glomeratus, for the control of cabbage butterfly, because once it is there in the cabbage, they devour or feed means voraciously. In 1762, the myna is introduced for the control of red locust in Mauritius, and it was successfully controlled. In 1888, that Escherichia purchasei, that cotton cushion scale, it has been controlled by use of the Wedelia beetle. This is one of the major successful in the history of USA so far. Next is 1898 in India, the coccinellid beetle. You can see here the coccinellid beetle. They have controlled the coffee green scale. And in 1937, the chalcid wasp, the Hymenopteron, it has been used in Kodur, and it has been brought from the North America for the control of apple woolly aphid, and it was successful. You can see here that small Aphelinus mali, they are parasitizing this woolly aphid. Even other successful examples, you can see here that control of woolly aphid has been controlled in Assam, the use of parasitoid, we call it Aphelinus mali. And another cotton cushion scale again, that Escherichia purchasei has been controlled in Nilgiris areas with this predator, we call it as this Wedelia. You can see here, this is Rhodolia cardinalis, which has been brought from the USA. Another successful attempt is also the use of the Sanjo scale, which is one of the pests in apples. It has been controlled by Chalcis parasitoid. Name is that Aphytis diaspidis, which has been again brought from the USA. The castor sibiluper, which is one of the pests sometimes occurs, and it has been we call it as Achia genata. It has been controlled by the use of another parasitoid named as Telenomus from the New Guinea. The pest of coconut, what we call it as rhinoceros beetle, it has been, you can see that it is a possibility of by the control by a predator which is known as Platymeris levicolis, and it has been successfully done, which has been brought from the Ginjabir. Now, the type of biological control agent, what we see is a predator, parasitoid and pathogens. In case of predator, they are free living organism through its life, and they are much bigger than compared to the host, and in their own one life cycle, they devour many more individuals of host insect. This is the predators, and in this predator, the immature as well as adult both stages attack, or you can say that devour the larvae. Here you can see that ladybird beetle, one is adult and another is this one is the immature stages, nymphal stages. They are it is devouring the aphids. The parasitoids are much smaller compared to the host, and generally in the immature stage it is parasitic in nature, whereas adult stage it is free living. So, when it enters into it in due course of time, within a very short period of time, it consumes the host, and they emerge out as free living. Here you can see that the Trichogramma, this is one egg parasitoid. This the insect, the adult stage, they lay egg inside within the egg. This is the beauty of the parasitoid, what we call it as Trichogramma. It is then it will parasitize, then egg will develop, and after at the formation of the adult, that the parasitoid will emerge out and search for another host. This is one of the, you can see that it is already commercialized, and Trichogramma is being sold in the case of Trichocard. Here you can see that in the pink portion that the parasitized fastidious host, we call it, it is being pasted there, and it could be placed in the field, and the small tiny this egg parasitoid Trichogramma will emerge and will search for new host. The third category of biological control agent is pathogen. These pathogens, they are microbial in nature, and it includes, as we say, that there are viruses, bacteria, nematodes, or even the fungi, or rickettsia it belongs. There are two types of this pathogen. One pathogen which the insect feed on it, it acts as the ingested microbe. When the pathogen enters into the body system of digestive systems of insect, then it starts their functions. So, this is known as the ingested microbe. Another category of the microbe or pathogen is that we call it as penetrating microbes. For example, here you can see that nematodes and fungi. It is from the outside, it acts as a contact poison. Here you see that bacteria, the examples, the most you can see that widely used in the pathogen in the bacteria category is Bacillus thuringiensis. It has been used for the control of Lepidopteran pest, and of course, we will come to in this. Then second is fungi. The species, what we call it as or genus which include is Beauveria and Metarhizium. The third category is viruses, and it is very specific, these viruses. We call it as such as, for example, here we have given two examples. One is Helicoverpa armigera nucleopolyhedrovirus. The second is Spodoptera litura nucleopolyhedrovirus. And the third category is that is granulovirus. In category of nematodes, these are different from the plant parasitic nematodes, which are harmful to the plants. These nematodes are entomopathogenic. They feed on, they kill and feed on the insects. The examples here we have listed here is the Stenonema and Heterorhabditis. The first that Dr. D. Herrell, it has used first biological control agent that we call it as Coccus Bacillus acridum for the control of locust. Then the Japanese scientist that Ishibata, he has isolated Bacillus soto, a letter which has been, you can say that which is from the larvae, we call it as Flachere. The similar organism has been isolated or from diseased floor moth, and the region from where it has been found out, it is thuringian. That is why the bacteria, it has been named as Bacillus thuringiensis. Another entomopathogenic bacteria which cause milky disease in white grubs. Here you can see that we call it as milky disease in Populia japonica. Here you can see this is adult Japanese beetle, and this is the entomopathogenic Populi bacteria, and this is healthy, and the infected the grubs. If we just, you can say, tease up, only the milky fluids will come out of it. Overall, if we see the categorize that the entomopathogenic bacteria, some are spore forming, some are non-spore forming. In the spore forming, we have obligate bacteria, and another is facultative. Obligate means they are very much associated or needed for its multiplication, whereas in case of facultative, it is if the main host is not available, then the alternate host, it will multiply. In case of non-spore forming, we have potential such as Pseudomonas and Aerobacter. In the facultative, we have Serratia. Various commercial products are available in case of Bacillus thuringiensis product. For example, you can see here dipel thuricide, bacticides, or Biotox. This variety, the BT Kostaki, it is being controlled for a large number of Lepidopteran pests, such as you can see that diamondback moth, bollworms, cotton prodinia, these are noctuids, and whereas Lepidopteran caterpillars. Other variety of this, that is Bt israelis, it is used especially for mosquito control. There are other variety like Bt bacillus thuringiensis, variety thuringiensis, it could be used for the control of flies. There are other variety which is known as Trebnionis, it is used for the control of weevils, and the last category is endotoxins, it could be used for the control of mites. So, here I would like to emphasize that how the bacteria acts on insect. What is the mode of action? So, first the caterpillars, because it acts as a stomach poison. So, when we spray in the field, so it is there on the leaf. So, when the insect, the caterpillars feed on it, then this caterpillars consume foliage treated with Bacillus thuringiensis. This the spores and crystal proteins, it will enter into the body system, means the digestive systems. Within a minute, the toxins that bind to specific receptor sites in the midgut region, and the caterpillars will stop feeding. And then within 1 hour, 2 hours or 3 hours, the gut wall will break down, and the content from the gut region will enter into the hemocell, and from the hemocell the content will come into the gut, and there will be, you can say that disturbance of homeostasis. Then within 1 or 2 days the caterpillars will die due to septicemia, that is blood poisoning, and the gut bacteria will then multiply and again it will disperse. So, here you can see that this is Bt crystal. When it is ingested, it is getting solubilized and then it becomes an active form. After the active form, it will just effect on bind to the protein of the receptors, target sites, and toxins lesions will occur, and the cell, that midgut cell, it will you can say that die, and it will create pores through which the gut content, you can say that will disturb. Another way of you can see that here the crystals and spores are there. When it is reaching in a certain pH, it will just dissociate and dissolve, and making this endotoxin into a protoxin, and then active toxin will act on the epithelial cells, and it will produce the perforations of the gut membrane, and later on this the spores will multiply or proliferate. In case of entomopathogenic fungi, more than 700 species of fungi are known which affect or infect insect. So, these are entomopathogenic fungi. Out of these 4 groups of fungus, the group which is associated with the entomopathogenic active, which we can multiply, then this is deuteromycetes. In this we have already commercialized products are available like Beauveria, Metrrhizium, Verticillium, Pneumoria, Pacillomyces, Aspergillus, and Histula. The first of all, Metrrhizium and Supplier, it has been used by the Russians in 1888 around, and it is used for the control of wheat chaffer, that is and as well as the sugar beet weevils. How it acts? There are 3 phases are there. The first, it will adhere to the surface of the insect, the cuticle. So, adhesion and the germination of conidia on the insect cuticle, and it will dissolve the cuticle. Then germ tube will penetrate into the interior portions, and then this in the hemocell, this fungal will dissolve and this hyphae will form and colonization will occur, leading to the death of host. Of course, it is not as fast as the bacteria, but it takes time. It takes about 5 to 7 days or 1 week it takes time in, means you can say that complete overcoming the immune system of the insect. Here you can see that first one spore is being attached, adhered to the cuticle. Then conidium is enzyme activity occurs and it secretes peptidases, chitinases and lipases, and it will dissolve the skins or cuticle. Then it will enter into the epidermal region and it will secrete enzymes and toxins that will, you can see that into the hemolymph, and the hyphal way or blastosphere space will be there, which will disperse and multiply further faster, I will say that. Here you can see that several this entomopathogenic bacteria, just we have named you, like Bacillus, this Beauveria bassiana is used for means, okay, we will talk about this one later. The Beauveria browniarti, then Metarhizium anisopliae, Nemoria raleigh, Verticillium lecani, Hirsutella thomsoni and Pasilomyces fumosoricus. Here you can see that the Beauveria bassiana, it is also known as white muscardine fungus because the spores produced is white in color, whereas the Metarhizium anisopliae, it produces the green spores, and that's why it is known as the green muscardine fungus. So these two are one of the main entomopathogenic fungus in the category. Here you can see that another very good entomopathogenic fungus is Beauveria browniarti. It looks whitish, but it is a very good product for the control of white grubs, which feeds on sugarcane roots like that. Here just we have mentioned that the category of 4 entomopathogenic fungus which affect the type of insect and what are the habitats. Here you can see that the Beauveria can infect on beetle, locust, caterpillars, termites, scaly insects and the aphids, and all they are generally the foliage. Of course, the termites they are subterranean region, but of course it can also act controlled by the Beauveria. The second category is the Metarhizium. They are suitable for the insect sucking insect pests. Here just we have listed that frog hoppers, beetles, even aphids it can be controlled, and of course the habitat is the foliage. The third category is Nemoria. It act or control or infect the caterpillars, the lepidopteran insect, and of course it is the habitat is the foliage. The last is Verticillium. It affects or controls the sucking insect pest such as the aphids, whitefly and scales. It can be used for the control of the insects which is living in, you can say that, or attacking the greenhouse or the foliage. Several products are available in case of entomopathogenic fungus such as Bassina, Biosoft, Biopowder, Beauverin. Whereas for in case of Metarhizium anisopliae, this is Biomax, Grub X and Metabase, etc. The one category of the entomopathogen is this entomopathogenic nematodes. You can see here these are the third stage infective juveniles which are searching a host. So what are the peculiarity of entomopathogenic nematodes? These are the nematodes which parasitize an insect and kill. So this is known as entomopathogenic nematodes. They persist in the soil in the third stage juvenile and they search a host by their physical and the chemical cues. Like they release, like host is releasing carbon dioxide or even the nitrogenous product. So that it will, through which it will search and it will find a host. This one of the, you can say that the peculiarity of these nematodes are that they are in symbiosis with one kind of bacteria, through which this bacteria, it is being used for the control of the insects. And more you can see here that the nematodes, the entomopathogenic nematodes, they are of two kinds, two genus. One is Heterorhabditis and another is Steinernema. In case of Steinernema, the bacteria which is associated symbiotically, we call it as Xenorhabdus, and it is being housed in a pouch. Whereas in case of Heterorhabditis, it is, the bacteria is name is Photorhabdus, and it is being, it is stick to the gut wall of the nematodes, and this is you can see that luminescent in nature. If you keep in the dark, it glows. So this bacteria, the Xenorhabdus and Photorhabdus, is being used for the killing of the insect. How it does that? This nematodes, I think this nematodes, it just, this nematodes, when after sensing enter into an insect, it will release bacteria. These bacteria in a certain specific pH level, it will just start multiplying. During the course of multiplication, it produces toxins, proteases and other enzymes which degrade the tissues of the host. Then these nematodes, they feed on the host tissues and continue for multiplication for 2-3 generations. And when it is the resources of the insect is being depleted, then the second stage juvenile, here the second stage juvenile, it will emerge as pre-juvenile stage, and selectively it will take the bacteria and put in their own stomach and then molt into the third stage and then migrate into the soil out of the host in search of another insects. So this is how the mechanism of action life cycle takes place. Here you can see that Steinernema, we can see it is the grayish color, and it is multiplication on the insect itself. And this the big one, what you can see is, this is known as the mother endotokia, in which the developing inside the this mother, the egg developing eggs are there, which feed the means internal tissues of the mother, and it will kill it. Here you can see the other side, that Heterorhabditis. Heterorhabditis, it looks like a brick red color, and it multiplies inside. It will never come outside the body. And after 1 week of completion, at a time when there is a moisture content, all juvenile will come out. So the life cycle event occurs in entomopathic nematodes are that first nematode will enter into the insect. It will sense that the environment is suitable, then accordingly it will release the bacteria. Bacteria will release the virulence factors, and then death of the host is occurred. In the third stage, the nematode reproduction occurs, in which the bacteria goes into the stationary phase, and production of antibiotics, exoenzymes, crystals, protein and bioconversion of the insect takes place. And in the last stage is that development of new juvenile, that is when the resources is depleted, the colonization of the intestine of infected juveniles occurs. So this is the events what occurs in case of here. Just I want to emphasize that in case of Heterorhabditis, if you are putting one nematode, then after the multiplication of 16 days, what we will get is about, after 2 weeks, we will get around 1,500 of nematode. In case of the Entomopathogenic, the second side you can say, 2 nematode is needed. This sexual reproduction is there in Entomopathogenic, whereas in case of heterodiabetes, it is hermaphrodite first stage. The host, what are the hosts which a nematode can infect? Then we can see here black man weevils, Japanese beetle, white grubs, then serrid fly, that is a dipteran, whereas black cutworm, that is lepidopteran, whereas the corn earworm, lepidopteran, and leaf miner, that is Liromyza. It could also be, as well as diamondback moth can be controlled. However, the most suitable or host for, or you can see that Helicoverpa armigera or Liromyza in controlled conditions, it will be good, it will be work. The another category whose is you can say that considered as a component of the pathogen, but the true potential is not utilizes these protozoans. They can infect, you can say that the locust or even the bollworms, even the coleoptera beetle, it can put. However, the research is going on in this category. So, overall what we have learned today is that what is the biological control agent, what are the component of component of integrated pest management, what are the advantages and disadvantages of biological control agent, and a small brief history of biological control agent, and you can see that means brief introductions to the subject like predators, parasitoids, and pathogen. So, overall what we can say that a sustainable way of managing pests requires that the growers must put trust in nature and give preference to natural method of pest control over the chemical control. Thank you very much.